This is the first video of Chapter 1. It goes along with Section 4.1, DNA and RNA. So overall, this whole chapter is about the relationship between DNA and genetics. Genetics are critically important for us to study when we're talking about anatomy and physiology because there are a lot of different diseases and disorders that are caused by genetics or that genetics contribute to. Understanding Mendelian genetics can help us find patterns of inheritance and predict how often a disease might occur in a given family. In this section, we'll be talking about the structure of DNA, how DNA is organized into chromosomes, and then comparing DNA to RNA. So way back in the mid to late 1800s, there was a Swiss biochemist, his name was Neisser, who was studying the nuclei of white blood cells. And this is really, really gross, but he was getting pus from bandages that had previously been on infected wounds. And he was looking at those nuclei in order to start to figure out what DNA was made of. By the early 1900s, we knew the components of DNA. We knew DNA was made of a sugar, a phosphate group, and a nitrogenous or nitrogen-containing base. But we didn't know what DNA was used for. We had no idea what role it played in the body. Then, in 1953, with Watson and Crick and their stealing of Rosalind Franklin's work, we knew the structure of DNA and also that DNA was the molecule of inheritance. So we've seen this picture before. DNA is made up of a nitrogenous or nitrogen-containing base. You guys can see the nitrogens here. A sugar, in the case of DNA, it's deoxyribose, and then a phosphate group. We actually have 5 different nitrogenous bases that can be a part of our nucleic acids. Okay, so we have double ring structures called purines. There are 2 of them. We have an adenine and a guanine. Okay, so A and G, you'll see those abbreviated very, very often. And then we have 3 pyrimidines. Okay, so the pyrimidines are cytosine, thymine, and uracil. And cytosine and thymine are in DNA. Cytosine and uracil are in RNA. So thymine is not found in RNA. So purines have 2 rings. Pyrimidines have only 1 ring. The overall shape of a DNA molecule is a double helix. I'm sure you guys have seen this before. If you can picture a rope ladder that's been twisted, that's probably the best visualization of a DNA molecule. The phosphate group and the sugar are on the outside, and then the bases are like the rungs of the ladder that go across and make up the steps of the ladder if you're visualizing a ladder. The nitrogenous bases pair in a very specific way, and there are 2 factors that play into how the bases pair. Okay, first of all, we need 1 purine with its 2-ring structure to pair with 1 pyrimidine, which is the 1-ring structure. So essentially, we need 3 rings to go across. We can't have 2 purines, it would be too wide. We can't have 2 pyrimidines, it would be too narrow. So we need 1 purine paired with 1 pyrimidine in order to give us a consistent width across the DNA molecule. Next, we need the right hydrogen bond pairing. So A, adenine, and T, thymine, they both have 2 spaces for hydrogen bonds. And one's a purine, one's a pyrimidine, so they always pair together. Then, cytosine and guanine have 3 spaces for hydrogen bonds. And one's a purine, one's a pyrimidine, so they always go together. So A and T always go together, C and G always go together. We hear the word gene a lot. but a lot of us have never heard an actual definition for the word gene. So a gene is one section of DNA that allows us to synthesize one protein. Okay, so a gene is a section of DNA that gives us one protein's directions. The genome is all of the genes a person has all together. So there's an analogy that I really like to use here. I like to think of a genome as an entire cookbook. Every recipe that I have ever made in my entire life, every recipe I will ever make in my entire life, everything from a roast turkey for Thanksgiving dinner to asparagus to chocolate chip cookies to a banana strawberry smoothie. Every recipe that I can think of is in my cookbook, and that represents my genome. It's all the recipes put together. A gene is one specific recipe. The recipe makes one thing. It makes one protein. Okay, so the gene is the directions. I can't eat a recipe for a roast turkey. I can't eat the recipe card. I can follow the directions and I can make the roast turkey and eat a roast turkey. Okay, so a gene is the directions and the protein is the product, like a recipe and the food it gives directions to make. Humans have about 20,000 genes. Most of our DNA is non-coding. It plays regulatory roles, or it's more structural. So we knew the shape of DNA back in 1953, and then in the early 60s, we were able to understand how that shape relates to its function. The actual X-ray data that was collected was done by Rosalind Franklin, and she died early at age 37. She didn't get any credit at all before she died for her work. Three men, Watson, Crick, and then Wilkins. We don't hear about Wilkins nearly as often, but they got the Nobel Prize using Rosalind Franklin's data. So now we're going to talk about the structure of DNA. Okay, so we know that DNA is a double helix. It's tiny, 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 but a very long molecule. In order to keep that double helix molecule from getting twisted and tangled and broken, it's wrapped around these things called nucleosomes that have a core particle, and the DNA wraps around these. And then these nucleosomes form into this zigzag pattern, and then those get looped even tighter, and then these tight loops get organized even more into what we see as a chromosome when we look through a microscope at cells in metaphase, in a very specific point in cell division. Okay, so we have this very organized way of storing our very long DNA molecules. Histones are another component of holding on to that DNA in an organized way and protecting it from tangling and breaking. Histones are proteins, and they're another way that the DNA can get wrapped around, kind of like thread around a spool, so that the DNA doesn't get broken or tangled. We can see chromosomes at some points during cell division if we look with the right type of microscope. We can even use just a light microscope to see the chromosomes when they're in metaphase, in the process of cell division. And we'll have what we call two sister chromatids that are held together at their center by something called a centromere, and they have proteins called kinetochores that attach together at the centromere and kind of pin the two sister chromatids together. So moving on to RNA. RNA is a little bit different. RNA is not the entire cookbook. RNA is just copying down one gene. Okay, so RNA happens gene by gene rather than copying the entire DNA molecule altogether. Okay, so an RNA molecule could be very short, less than 100 bases, or it could be really long if it's a larger gene. There are also 3 different types of RNA. Messenger RNA is the one that carries the code from DNA in the nucleus to the ribosomes in the cytosol. Ribosomal RNA is one of the 2 things that makes up the actual structure of a ribosome. So a ribosome is made of ribosomal RNA plus some proteins. And then transfer RNA is something that we use in the process of translation, in the process of making a protein. Transfer RNA, or tRNA, carries the individual amino acids to the ribosomes to be added to the growing protein. RNA is not a double helix, it's just one chain, and it doesn't have deoxyribose as its sugar. Instead, it has ribose, and we already talked before that RNA has uracil in the place of thymine.